But welcome everyone to today's joint Division of Carbohydrate meeting and GlycoNet meeting. It's my pleasure to introduce Nicola Pohl to this audience. She's zooming in from Indiana University in Bloomington, where she is the Joan and Marvin Carmack Chair in Bioorganic Chemistry at Indiana University. She's also currently the Associate Dean for Natural and Mathematical Sciences and Research in the Colleges of Arts and Sciences, in addition to running her lab, where she has been working on and being a real fixture in the glycoscience community on her efforts to develop synthetic methods for solution-based automated oligosaccharide synthesis. And I think she's going to share some of that with us today, both in terms of the flow-based methods that she's been developing, as well as open source software. In addition to all that, I just wanted to say from a personal note, Nikki is a wonderful mentor, and I've had the pleasure of interacting with her here when she was at Harvard as a Radcliffe Fellow as well. And so I hope that you'll be able to get to know some of her work now and through the networking session at the end of the seminar series. So Nikki, thank you for taking the time to share with us today your work, and we're looking forward to your talk.
Thank you very much. So given the fact this is a webinar and there was a substantial amount of time to talk at the end of this, I wanted to give a talk that was meant to be open-ended and thought-provoking. And how can we make glycan synthesis fair? And some of you may have heard of this idea of findable, accessible, interoperable, and reusable, but I realize that many of us in the chemistry community, this is still rather new. And so what do we mean? What is a fair culture in chemistry? So when we mean findable both by humans and machines. So in other words, are the experiments that I'm running here in Bloomington, Indiana, findable by anyone across the world? Not only by you as people doing this, finding these experiments, but also machines, search engines, different mechanisms that we use to be able to find information. And one of the things that's very clear that we need not just the data itself, but metadata. So in other words, if I gave you a whole bunch of numbers, which I know are NMR peaks, that they're going to be uninterpretable if I don't give you more information. So that would be metadata. It's not just the data itself, but it's also the metadata that can be read by machines and searched in some sort of form. Then they need to be accessible. So once you find it, you need to actually be able to get a hold of the data. Otherwise, the data is not very useful. And obviously, there might be authentication, authorization procedures, especially if we were talking about data related to human subjects. But we still need some ways of accessing the data. Then the other part of a fair culture in chemistry is interoperability. So we need to be able to integrate that data into larger data flows. And that is a part that we're just beginning to think about doing within the chemistry community. And then finally, we need this to be reusable. We need for the data and the metadata to be described well enough to be easily replicated or combined in a variety of contexts. And those of you who do synthetic chemistry know that it can be quite challenging sometimes to reproduce people's work. And frankly, it's just part of the culture when you're manually doing a lot of different reactions. And I didn't even think of it that odd until I was talking to an astronomy colleague as an assistant prof who couldn't believe that I could spend months trying to reproduce something from the literature and not once think fraud. I just thought, well, I'm not quite adding it at the right speed, or I don't have this or that. And eventually I'd get it reproduced. But the fact that all that information was not in the protocol itself was considered very odd, for example, to an astronomer. So why would we want to have fair data principles? Why do we want glycan synthesis to be fair? So a major part of this is public trust and reproducibility. So if you can easily reproduce data that we collect and generate, both positive data as well as negative data, that's going to help us in terms of reproducing our work from not just in Bloomington, but in Canada and Europe and Asia, everywhere across the world. And then also being able to easily share that data. So this is especially important to maintain the public's high regard of science, especially when we're perceived to have a reproducibility crisis. Neither of my parents are scientists, and I was amazed when, after an Economist article on reproducibility, they somehow got this sense that most of science wasn't particularly reproducible. And I had to assure them, no, that wasn't indeed the case. Most of the things in the literature can be reproduced. But obviously, this is a key portion of having public trust in science, is that it is easily and readily reproducible. Of course, reproducibility also saves a lot of time and frustration, and sometimes that can mean lives, that time saved. The other thing that I'm excited about in introducing and adopting fair data principles is that we can start getting insights into very complex problems. When you think about the undergraduate organic chemistry textbooks, we have a lot of information about how to, for example, differentiate between an alcohol and a ketone. But now when you have something like carbohydrate that has multiple alcohols, the textbooks are silent. The subtle differences between one hydroxyl and another, we don't really have a good handle on, except in very limited contexts. And so for chemistry to be in the position to start using other techniques like machine learning, artificial intelligence techniques that are developed to illuminate these complex problems, we need good, reliable, reproducible data sets. And that is part of fair data principles. And then finally, of course, training. Chemists who learn data science skills and who learn this ability to work within a fair data principle framework can transport those to other domains in a job market that really highly rewards these abilities right now. So then goes to the question, well, how fair is it right now? When you think about glycan synthesis, both the synthesis of the monomers that we need to chemically string together into oligosaccharides, how fair is it? If you think about findability, the vast majority of data is locked up in PDF files behind paywalls. It is not easily findable for the average person. And sometimes it can be hard to find even for the trained person. And then there's accessibility issues, especially with all the authentications and procedures and debates about which journals we can afford to pay for. So it's not only the accessibility of the actual data, the actual procedures, but then also the accessibility of how we produce those experiments. There's actually extremely little open source or automated methods in our field compared to a field like bioinformatics, for example. And then interoperability. The reason I have this round bottom is think about how annoying it would be if we didn't have the standard size ground glass joints. You know, you'd have a one stopcock or a distillation tower and you would only fit to that one flask. It would be so annoying because if you broke that one flask, it would be a major nightmare to get everything together. So we've managed to get standards for glassware so that they're interoperable. I can buy a round bottom from one company and a distillation tower from another company, and they all have a standard ground glass joint. Those are truly interoperable. But unfortunately, if you look at most automation platforms, for example, they're not interoperable. You can't just buy other parts from another company and just fit them right in. And the software that we use to run most of our equipment is also not easily shareable or interoperable, let alone the data that's produced by all these pieces of machinery. Those are also not in formats that easily can be integrated with other data sets and shared with one another. And that makes it hard for these to be reusable. The data is made and then it sits. We don't have net data and metadata on that data that are described well enough to be easily replicated and be reusable around the world. So how do we get to that? So can traditional methods of chemical synthesis ever be fair? And this is from a couple of years ago when people could still travel. I'd been alerted to the Apothecary Museum in Heidelberg, and I actually got to see Bunsen's actual Bunsen burner. And we still have this in the lab. We still have beautiful glassware. We still have heating baths and a lot of the traditional things that Bunsen would feel right at home at. But that does in part lend to the inability to very quickly reproduce things. So for example, in my own lab, I found that the rate of addition of a Lewis acid would affect the product distribution outcomes. And that is not generally a variable that we ever report, even in the very best of literature preps. We don't report all the different things because these are manual procedures. I don't know exactly what the rate is that I'm adding by syringe the component, and nor do I specify how long it takes me to get the next component added into the round bottom. But these are variables that can make a difference in the reactions. So if I want to have a fair culture in glycan synthesis, one, I need to start looking at automating different processes. Even if it's not an entirely automated workflow, each individual portion needs to be automated so that all the variables can be specified and thereby replicable around the world. And when I think about glycan structures specifically, I'm just looking for 3 to 9 mers strung together, because the binding pocket of an antibody or a protein is usually only interacting with a few sugars. It's not interacting with 100 sugars. It's interacting with 3 to 9 sugars. So if we can very easily get access to well-defined carbohydrate structures, this could be a lot of time savings and eliminate a lot of tedious labor. Because ultimately, we'd like to be able to generate diverse carbohydrate libraries for systematic structure-function relationships the way that we take for granted in making peptide libraries or oligonucleotide libraries. And given the incredible improvements in AI techniques and computational power, it would be nice to be able to develop readily reproducible chemistry so that we can get much larger data sets, so that we can start looking at AI prediction algorithms and getting a handle on how to think about these very subtle differences among hydroxyl groups. And of course, if you get a machine to do things, they can oftentimes work much faster than I can possibly syringe 2 things in at once, even though I'm quite good at it now. And so you can access reaction regimes that would be impossible to do by hand. So I presume most of you haven't really thought about, how do I automate biopolymer synthesis from scratch? So we'll do a little primer here, review. One of the things that we oftentimes think about is some sort of purification system if we're going to do multi-step synthesis. Now, so far, everything I've talked about with the fair synthesis doesn't necessarily need to be for a multi-step synthesis. This can also be fair principles for a single-step synthesis. But if we want to start thinking about biopolymer synthesis, we oftentimes would like to have some sort of handle that allows us to fish out the mixture from the entire growing polymer chain. Then we also need the actual building blocks to feed to this machine, or however we do this multi-step synthesis. And then, of course, we need the actual chemistry in order to attach the building blocks to one another and, for example, deblock the next reactive group. One thing to keep in mind when you're doing automated processes, whether they are on a solid phase or a solution phase tag, is that you need these components to be soluble so that you can easily deliver them through the automated liquid handling systems. There are some ways to deliver solids or slurries, but they tend to be much harder to get reproducible. And so you're more likely to get line clogging and other problems of reproducibly delivering the exact same amount. And so generally you want to think about, how do I develop chemistry where all the components remain in solution? So my lab for the last 15 years or so has been developing a process that's based on a soluble fluorous tag. So this little C8F17 tag in purple is a tag that allows this component, the sugar that it's attached to remain in solution in organic solvents throughout a cycle of this machine that we have programmed now to be able to do a wide variety of chemistries, both glycosylation and deprotection chemistries. Our platform is computer controlled, allows us to systematically and reproducibly add a certain amount of reagent using these liquid handling lines. We can cool and vortex the reaction mixtures at very precisely controlled speeds and times. And then we can purify the growing polymer chain through a fluorous solid phase extraction cartridge that allows us to fish out the C8F17 tag material from all the rest of the material. And over the course of the last 16 years or so, we've developed a lot of different C8F17 and other fluorous protecting groups, modified protecting groups that we can attach either at the anomeric center as drawn here, or at various other centers to be able to do this kind of iterative process in solution. And so we have routes to rhamnons, glucans, sulfated compounds, mannans, and manuronic acids, all developed on this ChemSpeed platform using this fluorous tag. And one of the exciting things for me as a chemist about automation is that it inspires the development of new chemistries. This idea of I need to have it be soluble, for example, gets you to think about processes in a different way. So for example, that was one thing that inspired us to develop a new non-toxic bismuth reagent that I have citations here below, but won't go into detail about, for glycosylation using sulfur anomeric tags. We've also worked with Clay Bennett's lab at Tufts to adapt standard manual processes to automation, and that required the discovery of a new additive in this particular case. So for example, the promoters given here, this hypervalent iodine species that Clay's lab has developed, we worked with him together in thinking about how can we adapt this to an automation platform. And what we found is that trying to use the procedures exactly as they had developed manually, we got not only the desired product, but a significant amount of the orthoester side product. Those of you who do glycosylation chemistry are familiar with this orthoester that rears its ugly head and can be a little bit challenging because it shares the same mass, and the NMR spectra don't look that different, but you can identify that. And the other problem that you sometimes see, and we would certainly see, is this trans-acylation reaction and acyl transfer. So if we had no base at all, we got a major amount of acyl transfer. If we use the pyridine bases that Clay's lab had reported before, we got significant amounts of orthoester side product in doing this reaction in a solvent that allowed us to maintain solubility of the promoters. And so through working with this and thinking about it some more, ultimately we end up going with a beta pinene as a different way of capturing the protons and thereby allow us to minimize the amount of these orthoester side products and acyl transfers. So it's not as simple as just, here's a manual process, let me just get the machine to do it. Sometimes by forcing the machine to have to only work with reagents in solution, which may change the concentrations, you then have to rethink the chemistry itself and make these kind of modifications. So the nice thing is we were able to find those, make those changes and then fully automate the process of activating these kinds of glycosyl donors to be able to go from these acceptors and build up this glucan chain. And we reported that a couple of years ago. More recently, Mallory Kern in my lab was starting to look at, well, how can we introduce modifications to glycans, specifically, for example, a sulfur linkage. The last thing I just talked about was activating that sulfur linkage, but the sulfur linkage is also oftentimes used as a way of making a glycan more stable to glycosidases, the enzymes that cleave glycosidic linkages. So by substituting a sulfur or a nitrogen or carbon, you can oftentimes shut down those pathways that might degrade the glycan of interest. The question is, how do we site selectively introduce such a modification in the process of doing an automated synthesis, especially when, as I just talked about, the two methods, they actually use sulfur activation as a way of doing the glycosylation. And so Mallory had the case of like, well, can we start using, for example, trichloroacidimidates and demonstrate a way of actually putting these compounds into an automation platform? Well, one thing she decided, first off, of course, you just try the procedures that we've always been using. And we have been using, which I didn't make very clear in the last couple of slides, the normal procedures, so-called. So in other words, the donor and the catalyst are added together to the solution of the nucleophile. Unfortunately, when she did it that way with this sulfur as a nucleophile, she ended up not getting as much of the desired product. She ended up getting side products again. And that inspired her to start looking at the so-called inverse procedure. Now, this would be something that essentially is impossible to do if you're doing this on a solid phase resin, but you can do that when you're talking about a solution phase approach. In this case, the acceptor and the catalyst are together, and now you're adding the glycosyl donor, as it's called. Once she implemented, reprogrammed the machine to be able to get this kind of inverse procedure to work on our automation platform, she now found that the major products were the desired products. She no longer found these funky orthoesters or these dimerization side products. She could now get the major products using these inverse procedures. So that's one thing that we really like about an automated solution phase oligosaccharide platform versus a solid phase platform is you can the order of addition of components can definitely alter reaction outcomes. And this is a variable that you can change in a solution phase process, which you can't really do in a solid phase process where you're consistently running reagents across the solid phase resin. And there's also a lot more flexibility in changing reaction conditions in solution than on a solid support. We can, in theory, and have in practice, use some solid phase reagents that you wouldn't be able to do if you were doing this on a solid phase. So we now have a wide variety of both inverse and normal procedures programmed into this automation platform, and we've made a wide variety of glycans. Unfortunately, I have to admit that most of oligosaccharide synthesis is not yet automated, because if you look into the labs of people doing our automated glycan synthesis, and manual glycan synthesis, the vast majority of the steps are involved in making the building blocks themselves. In other words, if you looked at this careful protecting group patterns that I showed you on those building blocks, it takes obviously multiple steps to make those. Could be 4 to 10, or in the extreme case, 14 steps to make the building block. And then it would be a relatively small amount to do the glycosylation deprotection steps. And so in the green here, this is the portion that has been automated by my lab and other labs around the world, but we haven't actually automated the way to make the building blocks. So most of our time is still spent doing a lot of manual chemistry to make in glassware with heating baths to make those building blocks. So how do we get around this problem? So when you think about the building blocks, it's a little bit of a different challenge. For one, you want to have diversity, and the other, you're doing this on much larger scale. So I haven't mentioned yet, but our solution phase process uses solution phase equivalents of building block equivalents. So we're using about 1.5 to 3 equivalents of the building block at each glycosylation step, as opposed to the 5 to 20 glycosylation building blocks, 5 to 20 equivalents of building blocks required on a solid phase to do this. Even so, even though we need orders of magnitude less building block, we still need building blocks. And so how do we deal with this? So how do we automate the synthesis of the building blocks? So all of the processes I've talked about so far have been in batch. In other words, we have a batch reactor where we add all our components. And this means if we want to do something on a larger scale, on our current automation platform, we will oftentimes just run things in parallel in the same that was a 16 parallel jacketed reactors, and we can always run, we can upscale by 16-fold just by putting the same amount in each batch reactor, and therefore we don't have to worry about scaling effects like heat management. That only goes up to a point. So if you look into industrial processes, especially in the petroleum petrochemical industry, the way this is done is by flow processes. These are continuous processes whereby your starting material goes through and your reactants are flowed through a reaction tube. And depending on how long you keep flowing things through that tube, that tells you how much of the product you get at the end. So it's a lot easier to scale because now you're just running it for a longer time. You don't have to re-optimize. Many of you have had to try to scale things up. You can get it to work on a small scale, but then you scale it to a different scale, whether it be smaller or larger, and you suddenly have to reinvent the reaction because now heat and mass management are no longer as easily reproducible. And so flow processes get around that. But now if you look at actual building block syntheses, how do we automate the individual steps to make a building block in flow versus batch? We have the challenge of the vast majority of the reactions that we use are using insoluble components. So for example, the one building block protecting group that the community can agree on is the so-called permanent protecting group, a benzyl group, drawn here. The standard way of adding that benzyl group to hydroxyls of a sugar are using sodium hydride or potassium hydroxide, for example. And these are biphasic reactions. They're not soluble. And so that was a big challenge when Keyvan decided to, okay, I really want to try to start developing flow reactions, but there's nothing published in the literature that I can do a benzylation in flow. And so he started looking into this. And I'm just showing you what actually worked using a solid barium oxide pellets as the base, and then he successfully flowed his components through a column of the barium oxide, and that allowed him to do these benzylation reactions inspired by something that David Bundel's group had done manually in a different way. So the nice thing is now you can scale this. The only problem we had, and this is a major problem for a lot of flow chemistry, is that the solid reagents that we use can start getting packed into the beds because they haven't really been designed to allow constant flow of a liquid through the solid matrix. And so this works, and it certainly, his new route allowed him to shorten the synthesis of this particular building block significantly over the 12 steps from glucose that it was the prior synthesis in the literature had done. So developing different ways of thinking about the building blocks can also shorten the syntheses, but we have still a ways to go in order to develop a full set of reactions. We then started collaborating with Clay Bennett's lab, and we hosted Sabara in our lab. at Bloomington back when we could still travel. We're still waiting, a backlog of people that I'd love to have host as soon as we're allowed to have them have visitors again. But together we ended up developing an alternative to these benzylation processes. And we're actually now developing a wide variety of flow reactions for all the different protecting group steps that you might need to build your building blocks, as well as concatenating these steps into multiple flow processes. And the nice thing about this is that you can start doing much faster reactions in flow, in part because of heat management, in part because we can also heat the reaction to hotter temperatures because we can essentially quench it very quickly by having it flow into the next reaction. And so now we have a nice process using KHNDS and DMF to do benzyl group additions, as well as we have reactions developed for not only acylations, but also siloprotecting groups, naphthyl, methyl, ethers, etc. Now we're working on actually applying these different methods and expanding them to a wide variety of different building blocks. So now we have automated flow processes for the building blocks. We have batch processes automated to do the oligosaccharide construction. I think if I actually could take a poll and ask how many of you are doing automated synthesis in your labs, almost none of you would say yes. So do we actually have an accessible reproducible process for glycan synthesis now? In other words, is it fair? And that's what I'm going to talk about in the last few slides. Is it actually fair? So it is reproducible. It's been actually quite nice to see because students graduate from my lab. Kevon's no longer in my lab. He's in Steve Towns' lab now as a postdoc, but we can recall the programs that he wrote to make the compounds he did. And it's actually much more easily reproducible than when we are doing the manual processes. The only drawback is these instruments are expensive. This is about $250,000. We have 3 of these in a row. We're one of the few labs in the planet that has 3 of these instruments. And to be fair, we have 3 of them so that we always have 1 or 2 actually running. These are not trivial to maintain. We have a fantastic machine shop and electronics shop at IU that helps us. We use a proprietary computer program. So if you have the exact same machine, we could easily send you the program and then it would be very easy to set up. But the reality is you might have a slightly different automation platform. And the code itself that we send you would have absolutely no information about the instrument itself to make it readily reproducible. So in theory, if you have the exact same machine, it is reproducible. But in practice, this is not accessible. So that's the question I've been working on the last 4 years is how do we actually make automated synthesis so accessible that we stop doing manual chemistry entirely? And so as part of my Radcliffe Institute project, I paired up with Benjamin Lee, who was an undergrad at Harvard at the time in computer science, and Alex Magellas, who was a grad student at MIT at the time. And we started developing an open source Python-based framework that allows me to control the pumps, for example, switching valves, all individually so that I can build any automated synthesizer. Because if you opened up the cover of an automated peptide or oligonucleotide synthesizer or an automated solid phase glycan synthesizer, all you would see is liquid handling lines run by pumps of various kinds and switching valves that allow you to change between reagents and then somewhere to collect. We're also working on putting together fraction collectors, for example. If you actually do want to capture the excess of your building block, you have a way of capturing it specifically. And the nice thing about this code is it allows us to be totally modular. And basically it's all controlled by this little $35 Raspberry Pi computer. So what this allows us to do is you can hook together liquid handling pumps that could be generalizable. These are just standard HPLC pumps. This program is called MechWolf. It's an anagram of FlowChem, invented by Benjamin, and the tubing. And we can actually tell you in the code itself exactly how you have to set this up. So how long is the tube that goes from the reagent to the pump? How long is the tubing from the pump to the heating bath in this case? And this is in flow, so the longer you run the reaction, the more compound you get. But of course, you can also have these all flowing, not into a flow setup, but also in theory, you could also set this up and specify everything going into a certain size round bottom flask to do batch reactions. This is what the components that form the apparatus. We allow it to, for example, give you the length, inner diameter, outer diameter. The Python code is meant to be human readable. So it's a higher level programming language so that you can actually read as a human being, oh yeah, the tube is 2 feet long with an inner diameter of 1/16 inch, etc. And then also spits out a diagram that shows you exactly how to connect all the components. So you know exactly how to reproduce what we build in our fume hood in Bloomington, in Cambridge, or in Alberta, or anywhere else on the planet. So now, our automation work is shared using PDF files supporting materials. And since I've been on the editorial advisory board for Organic Letters and JOC, I brought this up in our editorial advisory board meetings. Is this actually fair? You know, one of the challenges is you have a computer speaking and the human doesn't understand it, and vice versa, or the dog in this case. So how do we make sure that humans can talk to computers and the computers and the automation can talk back to us, and that these are truly shareable between them? Because the PDF files are essentially just human readable. And this is a bigger problem. This is from an article last year. Frankly, most chemistry data sleeps, as they call it. On the whole, the chemistry community doesn't have an inherent fair culture. So they're isolated exemplars, but they state quite accurately the vast majority of data generated are not utilized. It's necessary to move away from the current situation of databases, data silos, and supporting information locked in journal PDFs and make more underlying data, both experimental and simulated, more available. And that's where we need to go. So for that end, Gavin Stamper in the lab has been working on a Jupyter notebook template to build our own open source electronic notebook for synthesis. And we can use this for both manual syntheses as well as our automated syntheses, because the manual part allows us to put all the components in, stoichiometry tables. It also allows us to put these inchy key strings that allow a machine to search for that specific compound. So like the IUPAC nomenclature that we put here, we also put the inchy key that allows a computer to read what is this compound here. And then that allows you to search things. So we now have also programmed it in so that you can add TLC data. You can put, for example, it'll calculate RFs. You can put all your calculations within the Jupyter notebook, as well as the actual code that does the automation if you have an automated process. So we also have programmed in stoichiometry tables. You can export this as a PDF file to mimic a paper notebook. And the beauty is you can now have all these PDF files on your computer. And if you know what the compound's inchy key is, you can just search your computer for the inchy key and it'll pop up all the lab notebook pages that use that particular compound. So they're findable now. And all of this is open source. I'd be happy to send you the template so you can modify it for your own experiments. And that makes it accessible and potentially interoperable. So in other words, you don't need a specific computer or a specific operating system in order to open these Jupyter notebooks. So with that, how fair is glycan synthesis? So we now have a way of making things much more findable. And in the future, we see a GitHub repository as a way of putting these into spaces that we can all share easily, not only sharing the actual automation code, but actually all the background of what the instrumentation looks like to run that code. It should be a lot more accessible now with low-cost automation using interoperable physical components like pumps. And we now have interoperability because it doesn't matter exactly which pump you're using. You can just use the drivers for your pump and then build whatever you use. So I see these components as things that you mix and match just like you are with your glassware. And that work is ongoing to make the data that we produce also interoperable. And then finally, we now have ways of making procedures more reusable. We're still working on making sure that all the metadata is described well enough to be easily replicated, but eliminating manual processes is a huge part of making chemistry and what we do actually reusable. And now we need to work also, can the data, not just the physical components, be combined in a variety of different contexts? And of course, we are not able to, neither manually nor in automated fashion, make every single kind of cool glycan that you might think of. And so we're actively working both in our own lab with collaborators to work on procedures to make glycosaminoglycans, microbial, and as well as plant glycans of all kinds. And so with that, I just want to end with our Zoom group meeting shot. A fair culture requires humans and their creativity. And for that, I've been very grateful of working with my students, and especially for the support from the National Institute of Health and more recently the National Science Foundation to develop this kind of science. So with that, I thank you very much for your attention.